a research agricultural engineer with USDA's PBARC in Hilo. She received her PhD from University of Arizona and did postdoctoral training at UC Davis. Her research focuses on developing pre- and post-harvest technologies to improve the quality of tropical crops grown in the Pacific Basin. Today, Dr. Liang will present on AI in agricultural research at PBARC. Dr. Liang. And the mouse works, is that okay?
Yeah. All right. Thank you for the introduction, and thank you for having me. I'm really excited to be here, to be learning from all the experts in the room as well. So my name is Pei. I'm a research agricultural engineer with the USDA-ARS-PBARC Research Service at the Pacific Basin Agricultural Research Center in Hilo, Hawaii. So, ARS, Agricultural Research Service, is one of the 29 agencies under U.S. Department of Agriculture. ARS is the chief scientific in-house research agency for the United States Department of Agriculture, focusing on delivering scientific solutions to regional, national, and global agricultural challenges. ARS has over 90 locations across the nation, and PBARC, the Pacific Basin Agricultural Research Center, is the only one in the state of Hawaii. So at PBARC, we currently have about 25 scientists, 14 postdoctoral researchers, and 60 staff. And these staff include all of our technicians and all of our administrative staff. So given its location in Hawaii, PBARC's mission is really to develop diverse, sustainable agricultural systems and pest management programs in support of Hawaii, the Pacific Basin, and U.S. agriculture. So we're currently comprised of 3 different research units, Tropical Crop and Commodity Protection Research Unit, aka the bug unit. This is a unit that's mostly entomologists and a lot of biologists that work on pest management. We also work on post-harvest technologies and quarantine treatment for export markets. And the Tropical Plant Genetic Resource and Disease Research Unit. This is our plant unit that focuses a lot on plant genomics and molecular biology, plant breeding, as well as plant pathology. This is also the unit that manages one of the locations of USDA germplasms that focus on preserving genetic diversity for tropical crops. And lastly, we have the Tropical Pest Genetics and Molecular Biology Research Unit. They're all really long names. And even when you put them in acronym, they still don't make any sense. So I have to put it up there so I can remember their names. But this is our newest unit that focuses on insect genomics and molecular biology for pest insects. So today, I'm going to attempt to give an overview of all the research projects we have at PBARC that has the AI in its workflow. And a good group of them focuses on pest insect identification. So these identification not only includes taxonomy, but also phenotype identification or classification. So for example, here, the first example is a beetle identification. So these beetles are a pest for our macadamia nuts. But when we set the traps out there to assess the severity of a pest infestation, usually in a trap we'll have multiple species of beetles in one trap. And in order for us to develop an efficient pest management strategy for these insects, we have to be able to accurately know how much of these insects are in each species, right? Because they're all lumped together. So currently, and on top of that, these beetles are really small. They're only about 1 millimeter in size. So for us to collect the trap and identify these beetles, we have to dump them all out and put them under the scope and look at them one by one. Am I still too quiet? Look at that. I talk with my hands. Look at them one by one under the scope to ID and count them. And this is, again, this is very labor intensive and time consuming, and it's not something that everybody can do. Only, I think at the whole PBARC, we have maybe 3 technicians can do this accurately and efficiently, right? So we have this project, it's a collaborative project with another ARS location in New York and Cornell University to use their imaging robots to produce these really high resolution scope images of these beetles. And from there, we were able to develop the AI workflow based on machine vision to classify them into by their species and quantify and classify. So that's a taxonomy example, but we also have some that look at different phenotypes. The example here is to look at the mating status of the Mexican fruit flies. And there's a reason why. There's actually more than one reason why we want to look at their mating status. But I don't have time to go into that. But basically, when we have a female Mex fly, we would want to know if this is a virgin fly or it's been mated, and they can mate more than once. And so we want to know their mating status with reasons. But currently, how this is done is to take the female Mex fly and take their reproductive parts and squish them, and then look at the sperm packets that come out under the scope. So it's not very obvious, and when this is not my project, so when the scientist who was leading this project sent me the images, I have no idea what I'm looking at. They just look like a scope image. But apparently there are these smudges, so this is where the part that gets squished. And there's these smudges around it that is the sperm packets. And the idea is that with these images that we have taken under the scope, we're hoping to develop a workflow to look for these smudges and to identify a mated Mex fly. And also from there, we're hoping that we'll be able to quantify how many times they have been mated and the quality of the mating event with reasons. But in addition to these image-based insect identification, we also have some projects that use a spectroscopy-based method to classify these different insects or their phenotypes. So one example is up there, we're looking at using near-infrared spectra to classify or differentiate a sterile and non-sterile Mediterranean fruit flies. There's a reason to do that as well. But that's one example, and then we're not looking at images anymore. These are just the spectra, pure spectra of our insects. And that was when we started those really preliminary, so everything that's just like 3D printed and stick it over there. But I think we can make it a lot faster now, the identification a lot faster now. And here on the bottom, and that's actually the same example when we were looking at classifying the beetles by their species. Instead of using the scope images that we collected, we can also do a very similar thing with their near-infrared spectra using a hyperspectral imaging system. So, moving on from all the bugs and identification of the bugs. We're still on bugs. You're gonna see a lot of bugs. But our research biologist and also our coffee specialist, Dr. Melissa Johnson, she has been developing this app for the coffee grower to help them manage their farm as a grower, as a decision support tool. She basically look at 2 things with this app: coffee leaf rust, which is a coffee disease, and also coffee berry borer, that's a pest in coffee farms. So as part of the app, she also have an image-based algorithm for the growers to be able to take a picture of their trap and then use that to estimate the quantity of the coffee berry borer on their farm. And from there, the growers can then have a heat map that shows where the pest pressure is, and combine that with the pest management strategies that we already developed and the weather information. They are able to give us some kind of prediction or suggestion in terms of when and where the growers can be spraying for insecticide or fungicide for their plant to manage on their farm. And from there, she further developed this a different app. Was that all these coffee images kind of relevant to what they've just presented. So as they are going around the farm, they have taken all these pictures of the coffee plants, right? So there's flowers, there's berries, but there's also the leaves. And from there, they went in the intensive annotating process of all these images. They're able to develop this AI-driven app that integrates expert experience, machine vision, and field validation to provide grower a tool for them to just take a picture of their plant and look at the leaf. They're able to identify, oh, these are flowers and berries, which is pretty obvious, but they'll also be able to identify nutrient deficiency, different types of nutrient deficiency, and also up there, they're able to look at coffee leaf rust symptoms and identify it for you. And while we're on coffee, we also have our research horticulturist, Dr. Tracy Matsumoto. She has been working on these plant growth regulator treatments for the coffee plants. The coffee is really interesting. If you go out to pick coffee cherries, then you would know, like you look at a branch, and some cherries are ready, and they're bright red, they're all the way red, ready to pick. But then you also see some that's kind of past due, and you also see some that's below, they're not mature yet, or you see some that's completely green. And they're all on one branch. So this non-synchronized fruit development is a problem for our grower, well, that's just how coffee plants are. But it brings up a few issues for our growers, right? So if they're all ripening or maturing at different times, that increase the harvesting costs, because then you'll have pickers ready, or pickers in the field for longer. Throughout the year, throughout the harvesting season. And also the multiple pickings can do damage to our trees. And for all the berries that didn't get picked, due to multitude of issues, like if they didn't get picked and left in the field, it also serves as a breeding ground for coffee berry borers. So her project mainly focused on developing these plant growth regulator treatments, so they can go from a non-synchronized fruit development to having them ripen all at the same time. And while developing these treatments, she also worked on taking all the pictures, not just of the coffee cherries, she also took pictures of these flowers, because they're flowers before they become cherries. So by looking at the different stages of flower, and look at this picture, you can tell that it's not synchronized, because you can see on one branch there's some flower that's just opening and some that's already ready to go, right? So while developing the regulator treatments, she also had these images, and the idea is with these flower images, they would be able to provide a tool for the growers to predict first when to spray. There's a timing after some days before or after flowering. That's the timing for them to spray. And also they will be able to predict the harvest better to have pickers ready, which is a very big thing here and across the mainland too, but labor management is a big deal in Hawaii agriculture. So lastly, one of our research biologists, Dr. Sim, she's been working on this genomic analysis pipeline. She uses Deep Variant, which is a Google-developed product that look at genetic variants in kind of an image processing way. So what it did is that they would have the sequencing data, and which is one-dimensional, right? It's a line of numbers, well, a line of letters, but they will pile them up so they can then they make it kind of into an image, and it's a two-dimensional image, and then you can do a deep learning-based method to analyze these images to call out the genetic variants. And these variants often it could be linked to a specific trait like insecticide resistance, or they could use these traits or these variants to identify different species or different strains of insects. And the better the model, the better the variant calling, and the more accurate these traits and connections can be. Is that right? Ah, nice. I'm right on time. Well, this is my last slide. So, to summarize, I think, as you can see, we have a pretty wide range of projects. And some are closer to growers, some like we're developing tools, we're developing apps for growers to use. Some are more on the research side, for example, the genomic analysis pipeline. For the insect identification, they kind of teeter, it's part pest management, but also a lot of them inform regulatory decisions for invasive species or for export markets. So it's a wide range of research that we have at PBARC, and how I see AI within PBARC, it is definitely a tool that helps us deliver better science and more efficiently, to start integrate more and more in our daily workflow. What I am hoping to see for the years to come with AI advances is especially given the labor issue, the employment issues that we have across the board. I'm really hoping to see a more integrated AI system that can be for grower support, grower decision support, and for their labor management, and for networking, and for workforce development, all these sectors. And also a more streamlined workflow for our specialists who work as the bridge between science and applications. So,
we have some time. Could take a question for Dr. Pei. Anybody have a question?
Are there any tools under development right now to address the coconut rhinoceros beetle?
Coconut CRB is a little different because I think it's not considered as an agricultural pest, so then it doesn't necessarily fall under our big umbrella as a part of the Department of Ag. But I do know there are people who are working on projects, maybe not AI related, but we have our scientists developing sensors for tracking individual beetles to look at their movement and find their nests as a part of the management strategy. But yeah.
Interesting. One more question we have? We're all set. Okay, thank you so much.